Another webinar, and this is the first webinar for September. Thanks to Professor Souza for accepting our invitation. And Professor Souza is going to talk about the Cuatro, Cienegas, and this is the first webinar for September. One of the interesting sites in Mexico. Professor Souza, she did biology, genetics in University of Mexico, and then she moved to University of California, and then to Michigan State University. Then she joined the UNAM Institute of Ecology. She's a very, very senior scientist now. And she recently had an article in Science, and it was very interesting about the site in Mexico. I'm sure we have a lot of things to learn from Professor Souza. So, Professor Souza, it's over to you now.
Okay. So I'm delighted to be virtually in India. It's a country I always wanted to go, but now I'm just my mind is going, not me. So what you see here is the Cuatro Cienegas Valley that has the shape of a butterfly, but the center of the valley is this mountain. Let me get the pointer. This mountain is very important. As you can see, it has a shape that no many mountains have. Is the product of tectonics, and it has fire in the belly. And this mountain has stored the aquifer and what we believe the conditions of the oceans of the past, along with the microbes. And where's that mountain? That mountain is in Mexico, which is on the other side of the world than India. And here's the butterfly-shaped valley with that incredible mountain in the middle. And all around the mountain, we have these different hydrological systems that have different conditions. And Cuatro Cienegas is probably one of the most diverse places of the world, but it is the most diverse extreme environment of the world. Well, Cuatro Cienegas is amazingly diverse, especially for microbes, but also for fishes, for turtles, frogs, very diverse in spiders and dragonflies or diatoms. So it's a very diverse place, and it's the only one of astrobiology interest where there's a complex food web based on the stromatolites and microbial mats. And the reason of this diversity, paradoxically, is the very unbalanced stoichiometry of the place. There's very little phosphorus, and all the phosphorus is being recycled by life. So this is a mountain, and you can see there's a red thing here, and it's magma. And we have isotopic evidence that the magma is there and is active. However, these are marine soft sediment, and there's no there's no conflict, there's no eruptions of volcano or anything like that. The only thing that magma is doing is recycling the water from the surface to the depths of the mountain and then go back. And so the cycle of the water depends on the magma, just like in Yellowstone. And this is a very special place where two supercontinents open, Rodinia in the end of the Precambrian and Pangaea in the Jurassic. And we are going to talk about three sites. Churince, that no longer exists, that is here, pozas rojas, and Archaean domes that are here. So as you can see, they are on two sides of the mountain. This site is dominated by gypsum. That is the signature of the Jurassic. That's basically shells of snails of the Jurassic that made their shells of sulfate carbonate instead of calcium carbonate. And this site is calcium carbonate. And so here's the story of the tectonics of Earth. And here are two very important events that shape the history of life on Earth. The first one, and it's very important, at the end of the Precambrian, when the stromatolites were already all over the shores of the world because they are from shallow water, the stromatolites capture the CO2 of the atmosphere into the reefs. And when Rodinia opened, apparently in Cuatro Cienegas, it gave more shore to the stromatolites, so they made more shelves of reefs. and that precipitates the cryogenian. That means that the Earth froze due to the lack of CO2 in the atmosphere, well, the lower of CO2 in the atmosphere, and that made the ice cover the continent mass and the glaciers scratch the surface of those continents and liberate the most important element for life, phosphorus. Phosphorus was stored in the rocks and was not available for the organism. So when clean water full of oxygen, by then the cyanobacteria had been evolving for 3 billion years, or 2 at least, and were putting oxygen into the atmosphere, but not into the ocean for also a very long time. When the ice scratched the continent, the shore was full of water rich in phosphorus. And the algae that had evolved on top of the stromatolite finally got the ecological condition to grow. And so they grew like crazy and precipitate another glaciation because they stored this time CO2 in their bodies. And the second bout of glaciation of the cryogenian is called the Marinoan. And again, the ice scratched the rock. Again, it freed the phosphate from the rocks. And this time was the turn of the animals. And early animals at first just glided on top of the stromatolites, eating the surface of the stromatolites, but then evolution allowed them to evolve jaws, and they start to scratch the stromatolites and make tubes and make caverns, and releasing in that action the nitrogen and phosphorus that was stored for a very long time into the marine sediments. And so by releasing these nutrients to the water column, the algae were very happy to have nitrogen. They already had phosphorus, but not nitrogen, and start to grow in the water column. And that turned the ocean blue. Before that, before the Dicardian, the ocean was orange. And that's amazing because it was the activity of microbes and then of the first algae and the first animals that transformed the Earth forever. And then we have the Cambrian, and Cuatro Cienegas was still in the shore. It's in the southern tip of Laurentia, and Laurentia is a craton that is very stable along the history of life on Earth. And then it was just in the breakage of Pangaea. And then basically the Atlantic was born in Cuatro Cienegas. And then you have all the northern hemisphere migrating to the north and the southern hemisphere breaking up. There was what is called the Western Seaway that connected the Gulf of Mexico all the way to Canada. And that closed slowly by the uplifting of the North American continent. And with this uplifting, the Sierra Madre Oriental was born from the ocean also and isolated Cuatro Cienegas from the sea 14 million years ago. So most of its life, the microbes of these ancient sediments had lived in the ocean. And basically they haven't realized they are now in a desert. And how we know that? How we know such a sophisticated things without having geologists scratching rocks? We know it by molecular clock. So here we have all the bacillus of the world. There are around 1,000 species well known all around the world. And all the known bacilli are the ones that have black branches. But the ones in Cuatro Cienegas are the ones with blue branches. And we can see that all this clay is very old in this tree. And there's a lot of specks of blue here. So let's analyze carefully. These come from only one side, the blue side that is on the side of the gypsum in called Turince. So when we do molecular clock, we have that the bacillus from Churince are very old. They are very different to everything else, and they were part of the marine sediment. They were the witness of the animals making holes and oxygenating the surface of the shallow sea. And that's incredible because bacillus need oxygen to grow, and there was no oxic conditions in the ocean for a very, very, very long time till this Cryogene era occurred. So we have here the evidence that these very long branches correspond to the first radiation of bacillus in the Dicardium. And then we had all the shorter specks of bacillus only from Cuatro Cienegas, and they all migrated at the same time when Pangaea opened and the Atlantic was born. But bacillus is just a small part of the story. Here we have the diversity in a lagoon that no longer exists, and all the circles, if they are small, like bacillus, is because they represent less than 1% of the relative abundance. If they are orange, they are from sediment. They are blue. These, for example, these actinobacteria are very abundant in the water, and the gray ones are in the soil. So the diversity is very high, and many branches do not have any circle because they are part of the rare biosphere. And we are learning more and more that this rare biosphere is very important for the equilibrium of the communities. So the diversity is enormous. And that lagoon, we have 5,300 lineages that could be described with 16S, only of bacteria and not very well Archaea. And when we do the same for the virus, but only the virus of the water, we found that Cuatro Cienegas here is in 3 sites, Churince, La Becerra, that is a spring that is 5 kilometers away, and Pozas Rojas, the site we are going to talk right now, is the most diverse place on virus in the world, meaning that even in the water, we have more hosts for those viruses than anywhere else in the world. And that's quite amazing because most of these viruses are related to marine lineages of bacteria. And so let's explore the next site, that is Pozas Rojas. Pozas Rojas is a very floatant system. Here we have it in winter, so it has not evaporated the water. But in summer, it keeps only this small part wet and the rest evaporates. And so it fluctuates from very hot and dry to cold and wet. And this site has a big lagoon that here we call Los Hundidos, is this one, and all these little pozas around the lagoon. And what happened in this place that make us look more is that there was a hurricane in 2010 that transformed the extreme oligotrophic balance of this place that was 156 nitrogens per each phosphorus to 9 nitrogens per each phosphorus after the hurricane. So it transformed his oligotrophic nature by releasing all the nutrients that flowed. It was, all this part was flooded by the hurricane, so it became one lake, reaching the detritus of everything that carried with the water. And so the ponds isolate themselves back where they were. So we think there's a recharging of the aquifer in particular points. And let's see who lived there, who survived the hurricane, and this change in the stoichiometry. And what we found is that the big lagoon that we will have absorbed most of the water will be the most rich in kind of all the water around it. But no, it is very different to everything else. These are each one of the 9 little ponds, and each one is unique in the proportion of, these are the abundant phyla, but there's a proportion of rare phyla that are very important, and this is with Illumina. So what is this deeper lagoon doing? So when we do something called non-metric multidimensional scaling to see the relationship between Churince, that was the previous site I just described, pozas rojas. So Churince is black, is here. Pozas rojas is purple, and here we have a purple here. And the Mediterranean Ocean is yellow. And when we go from phylum to order, to family to genus, we see that Churince and the rest of pozas rojas clump together, but the deep lagoon doesn't. Deep lagoon has another source of water within that mountain that is different from the shallow little pozas. So that itself is very interesting, and we need to explore it with geophysics and to know the source of the water. We are going to do that, but COVID changed our plans. And this is the third site, and the most fascinating of them is called the Archaean domes. And you see this, it is a very shallow pond. It measures less than 50 meters per 20. And here's one of the iconic pozas, the Poza Suldos. And this place is totally different from this place. Why? This place is different to anyone that we have seen in our life. It makes flexible microbial mats that bubble inside the atmosphere of the past, and outside there's photosynthesis. So these elastic microbial mats have not been described before. So let's explore them a little bit more. So as you saw here in the poza, there's these orange spots that don't form in 4 years of exploration. They never had a microbial mat on top of them, and we don't know why. And there's the place with microbial mats. So a friend called Frederick Talasso with a field gas chromatographer measured when the domes were drying in April 2016 when we discovered them. And then when they were dry under the crust of salt in January 2017. And what he found is very, very interesting, because when the domes were drying, these two sites, well, one side had escaped, the methane has already escaped. This one was better stored in a crust in the drying dome. And this one is different. And it's different in April, and in January has more than double the methane content than site 1. Site 1 is where we sample always. Site 3 we have never sampled. And we are going to sample this October if everything works. And there was no detectable CO2 in these 3 samples in April, but all of them have enough CO2 to build up this methanogenesis in January. So yes, the Archaean atmosphere is recreated inside these domes and under the salty crust. So we have to stage wet and dry. Most of the year is dry, and only when there's heavy rains, it develops these amazing microbial mats. And so since we call them the Archaean domes, let's look for Archaea, because most of the year is extremely salty. And when it rains, it's also very salty. 50% of salinity is pretty high anyway. And we found with metagenomics, complete metagenomics, that there's a lot of bacteria. That's not a surprise. There's bacteria everywhere. There's lots of virus, especially when it is wet. And Archaea is the most diverse place of Archaea probably in the world. In one site, in one sample, we found 230 otus of Archaea. Of them, they are common, no matter if it's wet or dry or drying, 188 that are constantly there. And that includes many archaea that are related to magma, to deep hydrothermal vents. So that reaffirmates the finding by geophysics that there was an isotopic signal of magma. Well, there's also a bacterial signal for magma. And when we compare with NMDS, the diversity of Archaea genera with Shark Bay that has been very well sampled for many years, Churince microbial mats, and Archaean domes, we found that the Archaean domes are totally different. And when it's dry, Archaean dome dry and Churince dry, they kind of could be on the same cluster, but still they are different. But clearly, Archaean domes are something else, and we need to explore these more carefully. And so I have a student analyzing not 3 metagenomes, but 6, and now we are going to sample the 7th. And she observed from 2016 April, this was drying 1, this was wet, this dry, this is wet, dry, wet, that the samples from 2019 have much more Archaea than before, and we don't know why. And the diversity using the NCBI database with CAT is humongous. There's 17,000 species. But if we use RRL meta that only compares the reads that are associated to full assembled genomes, the diversity is much lower. And that is because there's probably most of the diversity in this place is unknown. And so here we have the mystery. And you can see we are here sampling our scale versus the scale of the pond. It is a very small pond. And the mystery of the orange circles that we were going to tackle in March, but now we hope to go in October. Let's hope that COVID goes down. And all our metagenomes come from here. And here we did a transect of 1.5 meters to see the heterogeneity of the microbial mats in this place, to see the spatial heterogeneity and its relation with the water, deep water. So we went to sample in October 2018 when it was wet, that's time 0. And from there, we derived a mesocosm called time 1A that lasts 5 months into the light chamber. And then we went back to the same place and sample in dry conditions. And the idea is to have a falcon tube that measures 5 centimeters in diameter every 10 centimeters. So we have 10 samples in 1.5 meters. And here you can see the scar that the sampling for the mesocosm did. And here are the falcon tube scars. So you can see that actually the microbial mat is like a fabric. And so time 0 is this sample, and 1A is this that got into the lab inside a jar, closed for 5 months. In time 0, we have that the diversity is higher, as expected, because it's a microbial mat. And what was not expected is that every 10 centimeters is unique. And what makes them more unique is the diversity of rare Archaea, or rare bacteria that are in the gray part. There's also a lot of fungi. It's very diverse in fungi. And the Archaea with the 16S primer that we use for Illumina is not so good picking up the primers. The Archaea as the full metagenome, but also each site looks different. And it's even more interesting when we check for the metabolome. That means all the organic compounds that are in each one of these 10 samples at 10 centimeters of distance. And they have 16,000 compounds. Each one of the sites is different and unique. No, 1,600 compounds. And when it's red, is that this has a lot of these compounds, but none other have these compounds. So each one of the samples is really metabolically unique. And we are going to do in the next sample more metagenomics, transcriptomics, and more metabolomics. But what is very interesting between the Archaea and the fungi, that they are less abundant, so we can do a Spearman correlation test to see who of them are co-occurring or not co-occurring. We found that the Archaea and the fungi are always together and they form a core that they are present all the time, while the most abundant bacteria seems to be fighting among each other. When they are green, they are co-absent. They cannot share the same space. But we have a group of mathematicians right now doing the web of interactions. And so far we know that it's not random and that it forms a big blob of 2,600 O2s that are making that big blob, and the other ones are not sharing. They are not part of the interaction. So we are analyzing those interactions carefully. And what happened with the jars and the mesocoque? Here you see the microbial mat with the chloroflexi and the purple sulfur bacteria that are the first photosynthesis, the ones where photosystem 1 and 2 evolve. And we have 10 flasks, and we can see that 7 of them were black. They evolved anoxic worlds, and only 3 of them evolved oxic conditions. They didn't smell as smelly, and they changed the color of the sediment to white, which was very interesting, and they are more green. And this one made a small blob. But they lost the flexibility of the microbial mat in the lab. So after 5 months of light and water, but not connection with the mountain, the diversity was quite high, but it changed. In the anoxic worlds, there was no cyanobacteria, and the oxic world, we had 2 cyanobacteria that are very resistant to extreme conditions. And this one, the one full of rare microbes, is the one that makes the tiny blob. So that makes us think that the connection with the mountain is very important, but also it is very important because the input of rare microbes. So when we went back and sampled in dry conditions, we found that cyanobacteria that are highly tolerant to salt and have low light chlorophyll were very abundant in all of them, but also the rare biosphere was most of the sample, and the diversity is near half under the crusty salt. But even though it's the most diverse site under a salt cover that has been described. So the diversity is very high, even if it's dry and under a salty crust. And probably there's some input of water, even if it's low, because we still have a lot of methane production when it's dry. So Archaea are very active when it is dry. So what's the hypothesis? The hypothesis is that this small pond that is different to all of them has more silt and clay, and we already test that, than the other sites. This has a little bit more sand, and this also a little bit more sand along with gypsum. And the size of the particle can be related to how well the water travels along these small particles. Clay is very hydrophilic. It stores water very well, and that's why we do clay pots. And then when you dry them, you lose most of the volume because most of it was water. And what we have seen is that the clay in San Marcos y Pinos has the same shape of the mountain. Somehow it was molded by the tectonics of the mountain and has a pyramid shape just like the mountain. And so when it rains, that is very rare. Most of the vegetation use that rain. But when it really, there's a hurricane, that rain gets into the aquifer and refills it. And also there's more ancient water that was from the glaciers in the glaciation time. And also, I don't think the marine water stay, but we know that the marine bacteria stay. And they stay stored into the rocks. And they survive because they are cycling up and down. They go up, they get fed by the sun, and there's more carbon and nitrogen because nitrogen fixing and carbon fixing. But then they go back down and they live out of minerals and magmatic energy, and then go up and change their life cycle. That's the hypothesis. And here's the mountain that stores the treasure of the past. And we need to drill a little bit further in order to go deeper. And the idea is in October, we are going to make a core of 1.5 meters. We have to start with something, and that's something we can deal with. And explore what is the heterogeneity between down and up, the surface and below, and see how different are each one of them, with transcriptomic, metagenomics, tags, isotopes, and all that we can put. And what we found actually is being shown just this year by the team of Morono, and they explore the South Pacific gyre, that is the site with less phosphorus also in the ocean. And these marine sediments are 20,000 feet deep in the sea. They extract the clay in cores, just like we are going to do, but with much less technology that they do this. And they found that 100 million years old bacteria survive pretty fine, just relaxed, not dividing. And when they find food, they grow. And when there's no food, they just stay put. They don't make spores. It's just that they are very, very slow metabolism for millions of years, waiting for the opportunity of food. And that's not so surprising, actually, if you think of it, because what life needs to survive is food in order to divide. And they can just hibernate and wait for the food to pass by. And most of life on Earth was a little bit like that, was a life without food, where the opportunity of food present by a dead something around. And the bacteria of Cuatro Cienegas, their favorite source of phosphorus is the DNA of foreigners, and they produce an enormous amount of antibiotics precisely because everybody that is not part of the community is part of the food system. And so that's why Mars and Cuatro Cienegas may be related in many points than one. Both had a shallow sea. And in both cases, the gypsum, in the case of the crater Gale, is because that gypsum was produced by the impact of an asteroid. And in the case of Cuatro Cienegas, it was the tectonic that put fire and sulfur on the sea. But we know that water is not in the surface of Mars, it's probably beneath the surface, and inside the new robots and also the mission of China and Europe is going to look below. And below, following the water, probably you will find some evidence of either past life or actual life. Because Mars breathes, it changed the amount of methane and sulfur in spring and summer than is different from winter and fall. So this change in the atmosphere content tells you that probably there's something still alive, but it is under the mountain, just like in Cuatro Cienegas, living out of minimal resources and waiting for the opportunity of something dead to eat and grow a little bit. But what can we extract from all these from the deep past? Here is a petroglyph from Cuatro Cienegas caves, where among the first Americans stepped over there and saw the aquifer and believed it was magic. This painting has 10,000 years old. And so let's go. In the Ibian, there was no water, hence no life, because Terra and Theia had just collided and the moon was just formed, and the conditions were too violent for anything to stay. But just after, as soon as the atmosphere started to form, as soon as the water vapor could stay and drip and rain, it rained the ocean. And it took 400 million years to rain the ocean. So as Darwin has put up, it probably started in a warm little pool where the conditions of phosphorus, my pet idea is that it was an asteroid impact crater where life began, because it gives you all the elements. The clay was the surface where the asteroid impact. Asteroids are rich in organic matter that was built up in the stars. And water could dry and up, and that helped the process of origins of life. But more important, the impact of the asteroid will free the phosphorus from the apatite mineral and allow that locally there was phosphorus. And so what we know is that life survived because we are here. And we know that there was a unique last ancestor because we all share the same machinery, the same replication machinery, the same genetic code. And so the question is, why so many species? What was the force of diversification? How species split into the tree that Darwin thought? And the answer is probably clonal diversification. So there's this big controversy between clonal evolution against horizontal gene transfer. And there's people that believe that humongous horizontal gene transfer was very important in the early beginnings. And I don't think so, in part because Cuatro Cienegas has told us that if there's no food, there's no sex and no travel. Basically because phosphorus is a very important element that needs to be stored, not shared. And so the genomes in Cuatro Cienegas are in general small. They have very few traces of horizontal gene transfer. And those few traces are always related to big steps in adaptation, probably because they were acquired by SOS repair systems. And also another thing is that horizontal gene transfer tends to homogenize the gene pools and slow diversification, while clonal diversification separates things very fast as one mutation away. And that's what Richard Lenski, my former mentor, did by evolving E. coli in a flask with low sugar for a very long time, 32 years right now. And at the 25,000 generation, there was a mutation that allowed a new way of using the sugar in the flask, and was a new species that can use saturated while the other ones could not. And that's very interesting because it tells you that clonal evolution actually can be pre-adaptive and faster than the other one. So this is the bacterial adaptive radiation as far as we know. And probably this enormous radiation occurred very early in the Archaean. Probably it started to occur in that warm little pool full of comet soup where there was a division of who will eat which source of carbon and how they will separate the sources of nitrogen and how they would grow in different ways. And I truly believe that since the beginning of life, it was cohesion of the community. It was mutualism that drove these communities to survive, no competition. Because if competition was very important in the early beginnings of life, you will end up with one dominant strain that could do everything, not with an enormous diversity of life. So cooperation and clonal diversity was probably the rule for survival on early Earth. And the result is this enormous tree of life. You have all the upper part, all these are bacteria. These are Archaea. And this is the eukaryotes, the things that we can see. And here is a branch that is very interesting called Loki and Thor, that we have them in Cuatro Cienegas. They have been observed in deep sediments, and they are believed to be the ancestral of the eukaryotes. So we need to dig deeper and look for more of these Archaea. They are a very interesting group of life that is not as diverse as bacteria, but have been neglected in the study of microbial ecology. And this is my vision of mutualism and eco-evolutionary feedback and cooperation in the building of this enormous diversity of the branches of the tree of life. We are just a tiny, tiny tip, but you have all these Archaea and all these bacteria that are sharing lots of different functions in communities that makes this planet fertile. They change the planet forever together. And I think that's a very important lesson of life on Earth. And here we have a microbial mat in the tip of my finger. And they are the most diverse communities that we know, but they are more diverse functionally. Here we have the methanogens down, the sulfur bacteria then, and here's the origin of photosynthesis, the purple and the green sulfur bacteria, and on top of them, the cyanobacteria and the diatoms, and here you have a tiny plant. And so we have the world functions in between my fingertips. And I think that's amazing. And as astrobiology goes on, I don't think in Mars there's a microbial mats like this. I don't think in Mars there was photosynthesis. Probably because there was not enough time for the evolution of photosynthesis in Mars, and there's no biosignal of it. And also they have a dimmer sun than us, and they have to hide inside a mountain. So I believe that in Mars we will have these layers, the kinolithotrophic layers that eat minerals and live very slowly. They don't have any rust. There's nobody coming and eating them up. So in Cuatro Cienegas, we have the lost world that survived everything. It survived the glaciations of the Earth many times. It survived the extinction of the animals 5 times. We are in the 6th one. But it's not surviving us, the humans of the 21st century. Here's my favorite site and the most studied site in Mexico. It died in summer 2016. But now is the best study place. 100 scientists studied it for 6 years. Everything possible, as fast as possible before it died. And why it died? It died because thirst of agriculture thirst. So the problem is that they cultivate alfalfa in the desert, and alfalfa is the most thirsty cultivar that we know. And here there are deep wells that irrigate this alfalfa day and night, but also there were canals that were built in the 70s. And one of these canals, the blue one, is called Saca Salada, and is drying up 90% of the wetland since it was created in the 70s. And so we are in the brink of changing this and give back the water of Cuatro Cienegas to Cuatro Cienegas in order to recharge the aquifer. Let's hope we can do it. But in order to do that, we need to change society, and we are changing society from the kids up. We have one of our enemies in the beginning was Lala, a huge dairy giant that now is our partner in conservation and is paying the environmental education through art from kindergarten to 6th grade. And these kids have a different consciousness of the environment after this exposition to art and creation. And for the high schoolers, we have a molecular lab where they are the example of the Nagoya Treaty. They are developing their own genetic resources, and they are understanding firsthand the enormous diversity and what is at stake if the water dries. And also these kids are the children of the poor farmers that live in Cuatro Cienegas. And they are explaining their parents and grandparents that we need to save the water. We need to treat the water as what it is. It's sacred. It's the link with life. So we are transforming society through their kids. Thank you very much.
Thank you, Dr. Souza. Thank you very much for an impressive talk. Now I'll read the questions to you.
Okay.
Okay. We have the questions in YouTube. So, right. So the first question is from Dr. Manan Desai. How accurate and reliable can molecular clock-based analysis be considered? Is this molecular clock model based on 16S RNA?
Well, for Bacillus, it was based on 16S RNA, and we use the most stringent conditions for this molecular clock, and I think it's very reliable because if we do this for Bacillus, it gives you one story. If you do it for others, it gives you a similar story. And no matter what you do with complete genomes or only with 16S, you obtain the same marine old signature. And we did it with 16S because there's not so many genomes of Bacillus, and we wanted to have a very good statistical approach of that molecular clock. But the paper is written and is in eLife 2018. You can check it carefully, please.
Okay, yeah, thank you. Then the next question is,
I cannot hear you.
shall I read the question once again?
Yes, please. I didn't hear it.
Okay. So the next question is from Patnaik. So far, there hasn't been any organism which has shown extreme resilience to perchlorates. How can Archaea survive in Mars when its surface is full of toxic perchlorates?
I don't think we will have Archaea in Mars. We will have something else. Archaea are the product of the evolution of life on Earth, not life in Mars. We don't have any idea, even if they have DNA in Mars. We know that it's possible that life evolved and that is lithotrophic way of life is possible because we have an excess of methane that cannot be explained chemically. But perchlorate is toxic to Earth life. We don't have any idea what is normal or not normal for Martian life. And there will be prokaryotes, no doubt, simple life with one membrane. But what inside those prokaryotes? No idea. They are the experiment we want to know. Did they have DNA also? There's only one recipe to store information, or there's many. For sure, they won't have the same genetic code that we use because that historical contingency only happened here. And I imagine that the origin of life is really easy. And maybe even in comets, there's origins of life that spurs and then die because when the comet gets close to the sun, the water evaporates, and when it goes far, it stores more ice crystal. water is the most abundant molecule on the universe. The thing is that life needs it, apparently, to be in liquid form. And so Mars had liquid water, and you can see the rivers and the beaches that those lakes left all around Mars topology. So water was there, and now probably water is still there, but inside. What evolved there? No, there's no biosignal here that can refer us to the events, the series of lucky events of Martian life.
Wow. Well, thank you. Then there's another question from Patnaik. I hope I can read this question correctly. Can we use rhizopheric soil bacteria to study their survival against Martian soil?
No, I think those experiments are futile because evolution took 4 billion years in order to survive in these conditions called Earth. And life transformed every molecule of the surface and probably a lot of the molecules in the deep biosphere. And the history is important. So if you put Martian soil with Earth bacteria, they have not known each other, and probably there will be minerals that those bacteria like, but there won't be any organic matter. And so the history of life in each place is essential. And we don't know the history of Martian soil. And Earth bacteria have not been in contact with that soil, at least since those planets separated a long time ago. Long time ago. Mm-hmm. The formation of the solar system.
Mm-hmm. Okay.
I can see a question about what's the size of a dome. They are this size. They are like they can reach 20 centimeters in diameter. They're pretty big, and they're amazing. They go from 5 centimeters to 20, and inside is recreating the atmosphere of the past. That's what is amazing. It is the niche construction of this community is building up either a bigger surface for photosynthesis for the outer layer and a larger living space for the anoxic bacteria. That's what amazing us.
Okay.
That's why we need to go deep.
Okay. So I've pasted the next question in the G chat. Maybe it's easy.
Yes, if I can see them.
Yes. So maybe you can read the question so that it will be in the video.
Where I can see the questions?
It's in the G chat. Where you saw in the G chat. In the G Meet, there is a chat box.
Okay.
Yeah.
What is the maximum size of the dome? Yeah. And Mars has been geological active in the past. That's true. Shouldn't be possible target of our investigation to lava tubes which are subsurface and could contain Martian microbial life, just like in the Pacific years? Of course. And I think this magmatic energy that's still there in Mars is not only in the past, it's still a core of Martian magma that could feed the energy for the rest of the things to occur. And I think what we found in Cuatro Cienegas, with much less equipment than the Japanese, and what they just found in the Pacific year, give us a glimpse of how resilient life is. Life is resilient, and given the chance, I think natural selection is the only force that is universal. But with what is playing that natural selection is what changed between planet and planet. Lava tubes are a great idea.
Okay. Right. Thank you. There's one more question. I'm just pasting it in your screen. What might be the reasons for slow metabolism in the old bacteria? from deep sea. What are the significance of slow metabolism in astrobiology? This is from
I think this is an extraordinarily important question. I think there was a hurry to grow up only when phosphorus came along in the cryogenia, because there was not enough energy to grow fast. So microbial mats have survived and stromatolites have survived in few sites, only in extreme places where life is slow. There's no algae to block them the sun. And they have very cohesive communities that grow very slow together. And that's very important because the survival of these creatures for a very long time depends on precisely not being in a hurry, only slow your metabolism till you find food and grow only where there's food. And so there's these feast and famine cycles. And if there's no food, we'll just stay put and survive. Hope that there's not enough mutation accumulating in your DNA to be yourself when you wake up. But it seems that most of the bacteria in the sub-ocean floor in the recent study in the Pacific show that, no, they become alive pretty fast. So it just needs the chance. And for astrobiology, we should look below the surface, look for very slow biosignatures, not for a blue sea. It will be very improbable to find another blue sea, because the blue sea in this case was a biosignature of fast-growing animals and plants. Before those fast-growing animals and plants, there was no blue sea. The sea was orange.
Oh, the sea was orange?
The sea was orange because it was very rich in sulfur.
Okay. And when you meant this slow growth, how slow was it? Is there any scale?
Well, there's no way to know if the ones in the Pacific year took 100 million years to wake up. There's no limit to how slow is slow. The only thing is that when you open your DNA, it can mutate, and that's the only, they will have a very slow molecular clock. It will be very interesting. That's why we want to make holes in Cuatro Cienegas and start to look for not only the rare biosphere and its source, but the deep biosphere.
Wow, yeah, thank you.
Well,
okay, there is another question.
Archaea, allophilic Archaea that grows in the lab takes a year to make a colony.
Okay.
So, and that's something that you can cultivate. Most of life you cannot cultivate.
Okay. Yeah, got it. Yeah, thank you.
My bet
another question.
That slow metabolism will be a rule in most of the life outside Earth. And there's another question. What studies have been done with Archaea ribosome for surviving extreme situation? I don't do those studies. You need to grow the bacteria in cultures and be a very careful molecular biologist. I'm an evolutionary ecologist. But there's lots of studying on Archaea ribosomes and on Archaea proteins and how they survive. My bet is that Archaea don't like the extreme conditions so much. It's just that they grow slow. And so in those extreme conditions, they are not outcompeted by fast growers. That's why they've been hiding in those extreme conditions. And that the adaptation to heat and salt and acid and everything comes afterwards. I think Archaea evolved in the same orange ocean as bacteria did. It's just that now they are cornered in harsher environments because they grow slow.
Okay, thank you. There's one more question from Surendra Vikram. It's on your screen.
Yes, life is easy, and it's an unknown. And life is easy just, we know that because there's the first biosignature of life. That means enriching carbon with small isotopes instead of heavy isotopes in a particle of graphite in a crystal of zirconium that is present in 3 rocks, one in Greenland, 2 in Canada, from the same 4.1 billion years ago. And so each rock is preserved in a different way, but each one has the same biosignature. The graphite was enriched by small carbon isotopes, not heavy isotopes. And that first signature occurred as fast as liquid water was present. And that's amazing. And probably there were zillions of origin of life experiments, 1 per pond or anything. It's just that one survived. That's the most interesting part, is that just one survived. And the mystery of why that one, we don't know. And the bigger mystery of all, and we have to accept that science has limits, is how DNA came up. We understand the world of RNA pretty much, but not how the jump from RNA to DNA and genetic code. It occurred everything at the same time.
Very interesting. Wow. Yeah, there is one more question from Amita.
The ocean behind the emergence of sulfur-based redox buffers? Yes, of course. In the early life, everything was anoxic. Oxygen was an anomaly that was probably linked to the clays associated to hydrothermal vents, because heat breaks the water and clays store the electrostatic signatures of the free oxygen there. But usually the sulfur cycle is the most diverse cycle, the most ramificated cycle, and the most unknown cycle. And sulfur is the key of microbial maths, because it's the one that makes the gradient, the redox gradient that allows coexistence between oxygenic cyanobacteria and anoxygenic methanogens. So yes, the key is sulfur. Sulfur and iron.
So when do you think, how do you think the enriched sulfur came?
How big what?
No, when do you think the rich sulfur environment started?
Since the beginning. Sulfur is part of the magmatic soup. Sulfur is essential part of the magma. So very early on, we know that Luca was an heterotroph, that he liked sugar, but also he survived sulfur because the environment was sulfurous.
Okay. Yeah. Thank you. Okay, there are no questions from our participants. I have a question. You're mostly talking about the viruses, right? In the Cienegas, you're talking about the viruses. So is that not dangerous when you're examining them?
No. These viruses are all viruses on Earth are very host specific, and they jump host, but they jump from mammalian species to another mammalian species, not from bacteria to us. They don't see us. And so all these deep biosphere viruses haven't seen a human in their whole life. They are only alive when they invade their host, and their host is a bacteria from the deep aquifer. So no problem with that.
Okay, nice. Okay, thank you. So I don't think there are any more questions. Yeah. So we'd like to thank you once again for such an impressive talk. This so much learning we had. Thank you very much. So our live streaming will stop now. Our colleague will turn it off. But we'd like to discuss with you a few more minutes in the Google Meet.
Okay. I'm ready.
Yeah, just a second. Yeah.